Hello everyone, I'm Mr. Furlong, and today we're talking signal transduction. In this process of signal transduction, we're going to take a look at how a molecule that might not be able to get inside of the cell can cause big influences inside of that cell. And it's a series of multiple steps that has to happen sequentially in order for us to get from point A to point B. So let's take a look at an example of one. One is with a molecule of epinephrine. What epinephrine does is it releases stored carbohydrates, which is in the form of glycogen in places like our muscles, and release it into our bloodstream. So we'll do this if our blood sugar levels are low or during times of activity when we need more sugar, we can get at that stored sugar inside of our cells. Now, epinephrine itself, though, doesn't actually get inside of the cell. What epinephrine will do is attach to a receptor. So the epinephrine is the ligand, and it attaches to a receptor, which causes a conformational change in a protein. This is what's called a G protein. This is what it really looks like, where we have the G protein that's embedded into the cell membrane. What you see there in green is the ligand that would attach to that receptor. The receptor is shown in blue. This conformational change causes the alpha subunit of the G protein to move over to an adjacent enzyme. Now this enzyme, adenylene cyclase, will take ATP, break it down into a molecule called CAMP, or cyclic AMP. Now AMP is just ATP, but two phosphates have been removed. So remember, ATP has three phosphates. If we remove one, it becomes ADP, adenosine diphosphate. And if we remove two, it becomes AMP, adenosine monophosphate. Now, these AMPs will attach to kinase enzymes that are in the cytoplasm of the cell. Now, this causes the kinase enzyme, which is inactive, to have another conformational change to cause them to become active. Now, as an enzyme, it will require a little bit of energy, so ATP is going to come into play, and now that kinase enzyme will split apart and now be active. In other words, it has phosphorylated. This active enzyme will now cause an enzyme called phosphorylase to become active. It previously was inactive, which will then cause a conformational change to make it active, and it heads on over to the glycogen molecule, which will break the glycogen into individual glucose molecules that can leave the cell and into the bloodstream so that we can now have a higher blood glucose level concentration. So this is just one example of a signal transduction pathway. We'll take a look at others in class, and I'll see you then.